Am I audible to all of you? Debojyoti,
Yes,
can you
ma'am.
please respond that
Yes,
whether
ma'am. Yes,
I am
ma'am.
audible?
Yes, you're audible.
ये ऑडियंस नाम प्ले करने की क्या है
तो
बोलून नाही राहिला काही
Debojyoti, kindly confirm me whether I am audible properly or not. Debojyoti,
Yes,
are you there?
You are audible, ma'am.
Okay, thank you.
Welcome.
Good
ma'am.
afternoon. Once again, we have joined the session for the third round or third technical session of the day. Let us move forward now. And the speaker for this session is pharmacist Utsav Nepal. Born and raised in Nepal, he is an experienced industrial pharmacist with a demonstrated history of working in the pharmaceutical industries of Nepal. He is skilled in analytical chemistry, pharmaceutics, good laboratory practice, good manufacturing practice, good documentation practice, and Six Sigma. His core areas of interest include drug discovery, pharmaceutical analysis, novel analytical method development, and method validation. He is an individual with over 7 years of experience in the pharmaceutical field and is currently working as the head of Quality Control Department as manager for Royal Sasa Nepal Pharmaceuticals Private Limited, Nepal. I welcome you, sir, for your session. Over to you, sir.
Hello, am I audible?
Yes, sir, you are.
Okay, good afternoon, everyone. First of all, I would like to thank ADAMAS University for providing me this opportunity to share my knowledge and experience to our viewers and community. I would like to extend my gratitude to all the dignitaries of ADAMAS University, all the guest speakers, and our viewers. So let's start with the technical slides. Is it visible?
Yes, sir, it is visible.
Okay. So, good laboratory practice. Whenever we talk about drug discovery and development process, laboratory stands as an inseparable part of it. Good laboratory practice, GLP, governs all the activities going inside a laboratory. GLP directly impacts how our research work proceeds inside a laboratory. So, what is GLP? GLP stands for Good Laboratory Practice. These are the rules, standard operating procedures, and practices established by an organization to ensure quality and correctness in the results produced by the laboratory. So, history of GLP. GLP was instituted in the US following cases of fraud generated by toxicology labs in data submitted to the FDA by pharmaceutical companies. Industrial Biotest Lab, that was the most notable case, where thousands of safety tests for chemical manufacturing were falsely claimed to have been performed or were so poor that the investigators could not piece together what work had been done, even though IBT superficially delivered the test results. So, these issues were made public in the hearings at US Congress, which led to FDA's publication of proposed regulations on GLP in 1976, with establishment of final rule in 1979, which is 21 CFR 58. The Environmental Protection Agency, EPA, had also encountered similar problems in data submitted to it and issued its own draft GLP regulations in 1979 and 1980, publishing the final rules in two separate parts. They are 40 CFR 160 and 40 CFR 792 in 1983. So after this industrial biotest lab scandal, FDA introduced the GLP principles. In 1981, Organization for Economic Cooperation and Development, OECD, based in France, produced GLP principles that are international standard. So I'll be going through those principles in the upcoming slides. The GLP principles. These 10 other major GLP principles. Number one, organization and management, facility, quality system, personnel, apparatus, materials and reagents, working procedure and SOPs, performance of the study, reporting of study results, storage and retention of records and materials, that is document control and safety. So let's move on with number one, organization and management. The laboratory or the organization of which it is a part must be an entity that is legally authorized to function and can be held legally responsible. So the laboratory must be a legally registered one. The laboratory must have managerial and technical personnel with the authority and resources needed to carry out their duties and to identify the occurrence of departures from the quality system or the procedures for performing tests and calibrations, validation, verification, and to initiate actions to prevent or minimize such departures. The laboratory or the management should ensure that its management and personnel are not subject to commercial, political, financial, or other pressures or conflicts of interest that may adversely affect the quality of their work. The laboratory must define, with the aid of organizational charts and organization management structure of the laboratory, its place in any parent organization, such as the ministry or the drug regulatory authority, and the relationships between management, technical operations, support services, and the quality system. The laboratory must specify the responsibility, authority, and interrelationships of all the personnel who manage, perform, or verify work which affects the quality of the test and calibrations, validations, or verifications. The laboratory must provide adequate supervision of staff, including the trainees, by the persons who are familiar with the test and or calibration, validation, verification methods, procedures, as well as the purpose and assessment of the results. Facility. The laboratory should be of a suitable size, construction, and location. The design of the laboratory should be such as to provide an adequate degree of separation of any activity which may interfere with the proper conduct of each study. So the facility must be large enough to perform all the activities without any hindrance. The laboratory should have sufficient number of rooms or areas to ensure that test systems are isolated from one another. The premises must have suitable testing and safety equipment. The necessary energy sources should be available. If the line voltage is variable, suitable voltage stabilizers should be installed. Storage rooms or areas should be available as needed for the supplies and materials and should be conveniently located. These rooms should be separated from those areas housing the test system or should provide adequate protection against infestation, contamination, or deterioration. Now, the next principle is quality system. Every laboratory management establishes, implements, and maintains a quality system appropriate to the scope of its activities, including the type, range, and volume of testing, calibration, validation, and verification activities that it undertakes. The laboratory management must describe its policies, systems, programs, procedures, and instructions to the extent necessary to enable the laboratory to assure the quality of the test results that it generates. The documentation used in the quality system must be communicated and available to and understood and implemented by appropriate personnel. So the elements of this quality system must be documented in a quality manual available to the laboratory personnel, which must be maintained and updated by a nominated responsible member of the laboratory personnel. If any regulatory authority comes for the audit, then the quality manual must be with the relevant personnel. So the next one is personnel. The laboratories must have sufficient personnel with the necessary education, training, technical knowledge, and experience for the assigned functions. They should be free from any conflict of interest and not subject to any pressure that would interfere with the quality of the results. The laboratory management must ensure the competence of all the persons operating specific equipment, instrument, or other devices who are performing the tests, calibrations, validations, or verifications. The laboratory must maintain current job description for managerial, technical, and key support personnel involved in tests and calibration, validation, verifications. So there must be a job description for every personnel working in the laboratory or the organization. The laboratory must also maintain records of all technical personnel, including those under the contract, describing their areas of competence, educational and professional qualifications, training, skills, and experience. So the next principle, it's apparatus, materials, and reagents. So all the equipments, instruments, and devices must be designed, constructed, adapted, located, calibrated, qualified, verified, and maintained as required by the operations to be carried out in the local environment. The user or the laboratory should purchase the equipment from an agent capable of providing full technical support and maintenance when necessary. So we are purchasing an equipment, it broke down. And we do have to call the engineers. Let's say they provide the service from out of the country. They are not available inside the country, then it will take time. So the user should purchase equipments from the agents that are capable of providing full technical support and maintenance when necessary. To ensure proper sampling and measurement, the laboratory must have required test equipments or the devices for the correct performance of the tests, calibration, validation, and verifications. So there must be all the required equipments to carry out the required verification or validations. So all the reagents, chemicals, including the solvents and materials used in the test and assays must be of appropriate quality. Reagents must be purchased from reputable manufacturers or dealers and be accompanied by certificate of analysis. In the preparation of reagents in the laboratory, the responsibility for this task must be clearly specified in the job description of the person assigned to carry it out. Prescribed procedures must be followed, which are in accordance with the published pharmacopoeial method or other standards wherever applicable. Records should be kept of the preparation as well as standardization of volumetric solutions. Laboratory must be able to show the preparation and standardization data whenever the regulatory authorities ask for it. So the next principle is SOPs, or the working procedures. So what is SOP? As standard operating procedure is a set of step-by-step instructions compiled by an organization to help workers carry out routine operations. SOPs aim to achieve efficiency, quality output, and uniformity of performance, while reducing the miscommunication and failure to comply with industry regulations. Every process in the laboratory should be performed in adherence to the SOP assigned for the individual process. The next principle is performance of the study. All the activities inside a laboratory must be performed as per the plan set by the laboratory head or in charge or any authorized personnel. The sample must be tested in accordance with the work plan of the laboratory after completion of the preliminary procedures. If this is not feasible, the reasons must be noted and the sample must be stored safely in the allocated place. Any deviation from the original work plan must be reported to the authorized personnel. And the study should be performed such that it does not interfere with the other activities going inside the laboratory. The next principle, it's the reporting of study results. Test results must be reviewed and where appropriate, evaluated statistically after completion of all the tests to determine whether they are mutually consistent and if they meet the specifications used. The evaluation should take into consideration the results of all the tests. Whenever doubtful results are obtained, they should be investigated. The complete testing procedure needs to be checked according to the internal quality system. The next principle is storage and retention of records and materials. Document control. Documentation is an essential part of the quality system. The laboratory must establish and maintain procedures to control and review all the documents that form a part of quality documentation. The laboratory must establish and maintain procedures for the identification, collection, indexing, retrieval, storage, maintenance, and disposal of all the quality documentation and technical records. All the original observations, calculations, and derived data, calibration, validation, or verification records, and final results must be retained on record for an appropriate period of time in accordance with the national regulations. The records for each test must contain sufficient information to permit the tests to be repeated. The records for each test, they must contain those information so that if the test is repeated by another personnel, they can repeat it easily. The records must include the identity of the personnel involved in sampling, preparation, and testing of the samples. And the records of the samples to be used in legal proceedings should be kept according to the legal requirements applicable to them. All records must be legible, readily retrievable, stored and retained, using facilities that provide a suitable environment that will prevent modification, damage, or deterioration or the loss of records. So they must be kept in well-secured places. The conditions under which all original records are stored must be such as to ensure their security and confidentiality. So that is a must. All the records must be kept securely. For the computers, automated tests, or calibration equipment, and the collection, processing, recording, reporting, storage, or retrieval of test calibration data, the laboratory must ensure that procedures are established and implemented for protecting the data integrity. Such procedures must include the measures to ensure that the integrity and the confidentiality of the data entry or collection and the storage, transmission, and processing of the data. The last principle is safety. General and specific safety instructions must be made available to each staff member and supplemented regularly as appropriate through written materials or poster displays or the audio-visual materials or the seminars. Safety data sets must be available to staff before the testing is carried out. So if our testing is about to start, the personnel must know what chemical they are going to handle so that they can take the precaution measures beforehand. Smoking, eating, and drinking in the laboratory must be prohibited. Staff must be familiar with the use of firefighting equipments, including the fire extinguishers, fire blankets, and the gas masks. Staff must wear the laboratory coats or other protective clothing, including the eye protection, wherever applicable. And special care must be taken as appropriate in handling highly potent or infectious or volatile substances. All the containers of chemicals must be fully labeled and include the warnings like the poison or the flammable or the radiation etc. wherever applicable. Adequate insulation and spark proofing must be provided for electrical wiring and equipments, including the refrigerators. Safety rules in handling the cylinders of compressed gases must be observed, and staff must be familiar with the relevant color identification codes. Staff must be aware of the need to avoid working alone in the laboratory unless otherwise it's very essential. And the first aid materials must be provided. Staff should be instructed in the first aid techniques, emergency care, and the use of antidotes in case of accidental poisoning. So those were the principles of GLP. So what are the objectives of GLP? The objectives of GLP are to prevent the human errors in the performance of the job, to prevent the equipment errors, to prevent unsafe and hazardous acts which could affect individuals or the property, to improve the efficiency of the performance, and to adopt good and safe SOPs, and recording systems. So I will be going on with this individually. So human errors. So what are the human errors? There are major three types of errors. One is the sampling error, another one is the storage error, and analytical method error. The sampling error, it occurs when we are not following the SOPs on sampling. We should use the sampling rods, or maybe scoop or spatula for the sampling, using the bare hands or without using the gloves. That might contaminate the samples. The other one is storage error. Some chemicals or some items, or let's say biologicals, are meant, if they are instructed to be kept in the refrigerator, then they should be stored in the refrigerator. Otherwise, they may get contaminated or destroyed. The other one is analytical method error. While doing the laboratory activities, let's say we had to follow the method A, or let's say we had to perform an analysis by HPLC. But instead of that, we selected another analytical method, which was not in the pharmacopoeia, without any proper justification. So that is the analytical method error. The next one is equipment errors. Equipment errors can be either calibration error, or the selection error, or the maintenance error. All the equipments, they must be calibrated timely, because uncalibrated instruments might give false results, which will ultimately affect the results that we are expecting from our test. The next one is selection errors, the selection of equipments. Some tests require a particular equipment or the particular instrument that must be used for that particular test. And the maintenance error, the preventive maintenance must be performed on all the laboratory equipments as and when applicable. So the next one is unsafe and hazardous acts. While performing in the laboratory, improper gowning, or the handling of corrosive chemicals, or the random chemical errors or procedure errors, these might be unsafe and hazardous. All the personnel working in the laboratory must wear the proper gowning, or the mask, hair nets or hair caps, or the eye protection, safety goggles whenever applicable. While handling the corrosive chemicals, gloves must be used. Random chemical errors. Let's say a particular test says use sodium bicarbonate, but we don't have sodium bicarbonate. So let's use sodium carbonate. No, that's not doable. Unless you can justify that sodium carbonate can replace sodium bicarbonate, you are not allowed to use sodium carbonate instead of sodium bicarbonate. So that's the random chemical error. So procedure error. Let's say a certain analysis has to follow from step A, B, C, and D. But we do A, C, B, and D. No, that's not allowed. If the pharmacopoeia or the SOP says you should follow A, B, C, and then D, then we should follow A, B, C, and D, unless we can justify the change in A, C, B, and D. If that is feasible, then we have to update the SOP. The next one is efficiency improvement. So how can the efficiency be improved? So we can do the time management or the manpower management, equipment calibration, and proper training. So manpower management, the workload division is very critical. We can divide the work among various personnel so that every work will be performed efficiently. All the instruments must be calibrated. There must be a set time frame so that the work will be done according to the time frame. And all the personnel who are assigned to do the certain task must have the proper training. That's how the efficiency can be improved. So, how does the GLP impact drug discovery and development process? So, whenever we talk about drug discovery, it has the direct link to laboratories. Drug discovery and development is a lengthy process which requires extensive laboratory work. And all the processes going inside a laboratory are governed by the GLP guidelines. That's why following GLP guidelines in research activities will ensure safety of the researchers as well as accuracy of the generated data. So, in any drug discovery or the development process, wherever the laboratory is involved, GMP is directly involved in it. Without the GLP guidelines, we cannot move forward in any laboratory work. So the GLP has the direct impact on the drug discovery and development process. So thank you.
Thank you, sir. As of now, can we request all the participants to put forward their questions in the chat box, or they can unmute themselves and have the questions directly to Mr. Utshav Nepal. Neha Rani, you have raised your hand. Are there any questions you want to ask regarding this session? Okay. Okay, so we find one question from Ankit Jaiswal. What are the difference between GLP and GMP?
GMP, that refers to good manufacturing practice. These guidelines, GMP guidelines, they are applicable to the manufacturing sites. And GLP, that is good laboratory practice. They are applicable to laboratories, that to the testing. So GMP, that's to the production section, and GLP, that's to the laboratory section.
Okay, thank you, sir. I hope your solution is resolved, Ankit Jaiswal. Do we have any other questions with us? Okay, another question we have, whether guidelines can be changed or not.
The GMP and GLP, these guidelines are set by the regulatory authorities. If they deem feasible to alter the guidelines, as we know that the technology is advancing day by day. So as per the current trend, these guidelines can be changed. But the regulatory authorities will change the guidelines. And we are to follow those guidelines.
Right. Okay, we have another question with us. Can we replace humans with machines in GLP for some extent to minimize errors?
Yeah, actually it's happening. It's happening in today's world. Most of the systems are now automated. It's happening, yeah. Humans are getting replaced by the machines in industries. It's happening.
Okay. We have another question. It was a follow-up question that any fixed time period is necessary for the changes in the guidelines of GLP and GMP.
Fixed. Sorry, I did not understand the question.
I would request Charanpreet Kaur to please write the full question. It would be easier for the speaker to answer your question in that way. Okay, we have another question. Is GLP change as per the countries?
GLP guidelines, we can either follow the US FDA or the WHO. Any regulatory guidelines we can follow. And yes, it depends on the particular industry, which guideline it intends to follow. But we can follow any guideline.
Okay. Sir, as I got the question, I think that Charunpreet Kaur was writing or trying to write that whether there should be any kind of particular time period that needs to be allotted for changes that needs to be done in case of the GLP or GMP. Is there any particular time limit within which we have to?
No, there is no particular time limit. The regulatory authority can alter the guideline as they deem it feasible.
We have another question. Why Ayurvedic medicine is so substandard and why government has allowed these without having any strong regulatory agency?
I'm so sorry, but I have very little idea about the Ayurvedic side. I'm from the allopathic medicine. So I could not answer that question, particularly regarding the Ayurvedic medicine.
Okay, moving on to the next question. It's again from Charunpreet Kaur. He asked that, like, once the guidelines are changed regarding a particular purpose, can they be changed again? I think this question was answered by a speaker.
Yeah,
Yes,
they
it's a
can be
repeated
changed.
question.
Yeah, they can be changed.
Right. We have another question. How are GLP studies monitored and audited by the regulatory authorities?
Yeah, we can hear frequently that some companies are facing the US FDA audits. So that's how the regulatory authorities send the dignitaries to audit the pharmaceutical or any laboratories. And they have a checklist which they will see for the guidelines. So they will see if all the activities in the laboratory are performed as per the checklist or not. So that's how they will audit any pharmaceutical or laboratory.
Okay, thank you, sir. We have one similar question from two different participants, like, is GLP regulating the Ayurvedic medicines nowadays?
Sorry, that's what I said. I don't have much
Exactly.
idea in Ayurvedic.
Okay, sir. Moving forward to the next question. Is GLP changing for industries or academics?
GLP is mostly adapted by the industries because that's where most of the audits are taking places. But I personally think that academics should also follow the GLP guidelines strictly.
Madam has placed a question. GLP for change medical academics. This question is not clear to me to ask, sir. So I would request her to please write down the question properly. Place the question properly. Okay. As of now, I can see that there are no more questions, and I think that this session was quite fruitful, sir. We intend to see you sometime in our university as well as we progress with other conferences and seminars in near future. So we have another question here. Why the Indian country industries are not keenly following the GLP rules?
Is there any reference that the claim is made that Indian country industries are not following the GLP rules? So we cannot blame the industries without proper evidence that they are following or not following. Because the data should be with the authority. If they know that the GLP guidelines are being followed or not. So they are doing the periodic audits, GLP audits, or let's say GMP audits. So they have all the data.
Yes. So we have come to the end of this session, and I can see no other questions being put forward to our speaker. And it was being written by the participants themselves that they have found this session to be a very informative one. And exactly, sir, the same thing goes from my side as well on behalf of Adamas University, that it was actually an informative session. Thank you, sir, for taking out your time and accepting our invitation and joining. So now I would request Mr. Debojyoti Haldar to kindly present a token of appreciation to sir on behalf of Adamas University. Debojyoti, I would request to share your screen. This is for you, sir.
Yeah.
Wish
Thank-
and hope that you like it.
Yeah,
Thank you, sir.
Thank you. Thank you. Thank you for providing me this opportunity. Thank you very
Thank
much.
you, sir. Thank you, Debojyoti. We have come to the end of all the 4 technical sessions right now, and this is the last technical session which is remaining. And the speaker for this session will be Dr. Sharad Vist. And Dr. Sharad Vist is currently working as an assistant professor in Faculty of Pharmacy, Tishk International University, Iraq. He is doctorate in pharmaceutical sciences and having 16 years of experience in higher education in different institutes and universities. He has co-supervised many PhD theses, supervised Masters of Pharmacy theses, and supervised 9 CSIR stipend projects. He has filed 9 patents, and he is right now the peer reviewer of Publon's Drug Research, Drug Delivery, and Translational Research, International Journal of Peptide Research and Therapeutics, ACS Food Science and Technology. So now, let me take the privilege of welcoming you, sir. Over to you, sir.
Share application. Share. Am I audible? Can you please confirm? Hello?
Yes, sir. You are audible.
Hello, sir. Thank you, ma'am. So,
Thank you, sir.
good afternoon, everyone. Our chairperson, vice chancellor, sir, and faculties and other dignitaries. So, my topic is on discovery of the lead molecule. I will cover the following aspects in the drug discovery. There are different type of source of the drug. Then some drugs are developed accidentally, which is the scientists were not working on it, but suddenly they developed, or they found some results and they develop the molecule. So then we cover the lead compound and its development. Then Lipinski rule. It is very important whenever we design any drug molecule. Then there are several methods to identify the lead compound based on the site of the drug action and the drug targets. Then we will cover the computer-aided drug design or in silico design. Then combinatorial synthesis that we generally do in the 96-well plate. Then we will discuss the drug discovery cycle. Then we will discuss a software. Then for the pharmacy, how we can use these tools for the development of our research projects also. I will discuss this and some software and website that are very useful. So this first one is just for a while. Okay. So source of the drug will be natural. In the natural, it will be herbal, bacterial, fungal, or animal source. It will be semi-synthetic, mean to say we are isolating it from the herbal source, and then we are synthesizing into certain molecules like addition and deletion of groups or maybe some bonds. Or it may be a synthetic that we generally synthesize in the industry. So in herbal drugs, for example, the Glycesa glabra, it contains glycetinic acid. It is used for the ulcer also, and it has several activities like anti-cancer, antibacterial, antifungal. The curcuma longa, it is also used for anti-cancer, for antibacterial and other activities. Both are herbal derived molecules. Some drug molecules are derived from the bacteria like streptomycin, tetracycline, and the erythromycin. So these are the some you can say the bacteria from which we obtain it, and what is the purpose or what is the class. There are certain fungal derived you can say antibiotics also, or it may be animal derived like lipase and enzymes, vaccines, and toxoids. Just those are generally used. Then we come to the semi-synthetic. For example, the natural alkaloid, camphorticin. So it is obtained from the herb. Then we can add or delete certain groups, or we can add certain we can say bonds also. By modification, we are trying to convert into a derivative, and that derivative is termed as semi-synthetic derivative. Generally, this natural alkaloid do not have a very good efficiency maybe for the receptor or the target site. But once we change its group, or we can say basically we are changing the hydrophilic hydrophobic behavior or certain groups at the receptor site and at the ligand side. If interaction is higher, so more the drug will or ligand will bind more efficiently to a particular target site. It will not go to other sites. For example, when you take paracetamol, it will just go to the specific receptor. Like in case of etoricoxib, it is a specific COX-2 inhibitor. So it will go to that side only. Similar in case of semi-synthetic, the another example is glycetinic acid. Carbamazepine sodium is a semi-synthetic derivative. It is used for the ulceration, which is a treatment of the ulcer. Then we use some synthetic drugs. There, as you know, the land is also fixed, a particular area. Then the cultivation is also a trouble. So if we can derive any molecule by semi-synthetic procedure is good, because as per the herbal resources, we can't meet to the all demands of the human. So we synthesize some of the molecules. So for example, this one, menthol. Menthol is used in the cough products and other purpose also, is even for the permeation enhancer also. In the pharmaceutical we work on it. So we are synthesizing the molecule in the lab. It is not obtained from the herbal resource. So these are the synthetic drugs. So now we discuss first the accidental delivery of penicillin, because it is the very common example. Even when we were a student in 2006, I think, yeah, 2004, 5. So we also discussed these things. So first in 1928, Alexander Fleming, he discovered he find that some mold is there that inhibit the growth of Staphylococcus bacteria, and then they isolate some component and term or name the component as a penicillin. So there are various classes of penicillin. Some are natural and others are the semi-synthetic derivatives. So these are the structure of different type of the penicillin. What is the common here is the nucleus. Actually, beta-lactam nucleus is common. Just they are changing the groups. If they change the group, so it's ADME, absorption, distribution, metabolism, even excretion or toxicity, permeability. These can be we can say altered. Altered means to say increase and decrease both. If I quote one example like this one, tetracycline and chlortetracycline, if we talk about, so chlortetracycline have a high permeation as compared to tetracycline, because due to the chlorine, it will permeate faster. So in this case, because we are talking about the penicillin, we are just modifying the various groups, and depending on the various groups modification, there is a change in ADME process. So here we compare, for example, the green one is the parenteral and the oral. It is a natural molecule, and other red one background, these are the semi-synthetic or semi-synthetic molecule. If we talk about the if we compare the protein binding, suppose, so there's a variation in the protein binding, or metabolism is also varying because the structure is changing as per the ADME is also affecting. So if we can say change the groups, whether hydrophilic group, hydrophobic group, or any counter ion, all these things will affect the drug molecule. So the basic thing was the actual molecule was not so efficient that it can bind to a specific target site. But once we alter it by different methods, then we can make a optimum compound structure that have a best efficiency or whatever we want, that type of property. If we want to increase the permeability, so we can design like this, is in terms of toxicity, absorption, distribution. So we can change these type of properties depending on the groups added or deleted. So now we come to this lead compound is there and its development. So a lead compound is arise from a variety of different sources, as we discussed. So you can randomly select a plant given, or maybe any animal or any resource. The drug discovery's lead compound is suboptimal structure. So as we discussed, it is a suboptimal. The component do not have a very specific binding property to a particular site. But if we change the molecule, it will we can say optimize in the structure also, and the permeability or biological activity or pharmacological or therapeutic activity will increase or alter. Actually, generally we use a term alter because increase and decrease both are there. So as it bears, which the lead compound bears suboptimal structure, some structural modification are required to improve potency, selectivity, and improve the ADME process. Further, if we talk, the chemical modification lead to is required to test biological and clinical effectiveness of drug. If we synthesize any molecule in the lab, so synthesis may be based on two things. First is we have some to say in silico methods. We are using computer aided drug designing, or we are using a herbal component. Then we can check its biological activity, microbial, or even the anti-ulcer activity or memory enhancing activity. These type of activities we can run. The lead compound are found by characterizing natural product, employing combinatorial synthesis, or by molecular modeling in a rational drug design. So in case this combinatorial synthesis, we prepare, we use a 96 well, where we synthesize 96 compound in a very less time, a least time we can say. So by this method, mean to say combinatorial synthesis, we can make a library of compounds. It is the easiest method also, but if we compare it with the molecular modeling, certain time it happen, we are working on a molecular modeling, it is giving a promising activity, but it will not show the activity. That mean to say because there are some exceptions also, like if we talk about the number of carbon, as we know, if number of carbon increase, the hydrophilic behavior decrease. But there is an example, methanol and ethanol and propanol. Methanol have 1 carbon, ethanol have a 2, and propanol have 3. While this ethanol have a more, we can say some fraction, or we can say solubilized with the water. So that's why it is very important, whether you are doing molecular modeling, you have to do finally a synthesis and confirm by the practical results, not by the theoretical results. So chemicals identified as hit through high throughput screening may also become a lead compound. So for example, we can induce a site directed addition or deletion, like we know the nitration, if it's a benzene, it may be orthometa-para, depending on the concentration we are using, or if we are utilizing any specific procedure for a specific site substitution, so we can change the compound. Once a lead compound is selected, it must go under the lead optimization. Lead optimization again is addition and deletion of the groups. Maybe on the molecule, some groups are not required for activity, and some groups are essential. So there are essential and non-essential groups on the molecule also. It involves making a drug like me to say, it will have a good affinity to a particular site and it will act as a drug. We also follow the Lipinski 5 rule also when we prepare a molecule or select a molecule. A suitable target for rational drug design must be selected on the basis of biological pushability and identified through the screening potential lead compounds against multiple targets. If we are either synthesizing or we are obtaining this molecule from any source, we can run any type of, we can say, evaluation. Or if we know about the nucleus that is present and we know this nucleus have this property, so we can run that particular type of experiments also. Like in case of glycetinic acid, glycetinic has a chalcone moiety, and chalcone moiety is a flavonoid and triterpenoid moiety, and it is a promising candidate for anti-cancer, antibacterial, and antioxidant activity. So in any other chemical, if calcium moiety is there, so we will use this for a particular specific type of we can say evaluation. For example, if we know the calcium will not give anti-inflammatory, so we will not test for the anti-inflammatory. But the thing is, if we change the molecule, we can. But again, it depends on how the person, or we take this example. For example, curcumin. Once I prepare ester of the curcumin and the glycyrinic acid, it combined together, and its solubility altered. But the thing was, it was not checked for the activity due to the lack of fund. Main thing is this: we can select any drug molecule by any source, but if we have any idea about, for example, plant is there, this plant belong to which family, then what are the different chemical constituents present in this particular plant? If we can say segregate those molecules or phytochemical constituent in a different what we say solvents, that is for successive extraction, then we can confirm the activity, and it is a better method for the academics. So drug libraries are often tested for high throughput screening, means active compounds are designated as HIT. So which can screen compound for their ability to inhibit, like a antagonist or as an agonist. So there is a Lipinski 5 rule. It is important while we select any drug molecule. Especially criteria is the pharmacological and biological activity depends on this, or may affect by this. Methods. Hello, am I audible? Hello.
Yes, sir, you are. You are audible.
Thank you. So there are some methods for identification of the lead. So random selection from the plant or an activity. Even we have a literature. From the literature, if we say the Solanaceae family have this component, and generally all the components have a same nucleus, so we can select a new plant for the same activity. If we modify, then we can choose that molecule for other activity also. In combinatorial synthesis, we already discussed, we prepare different libraries of the chemical in 96 well. In silico or computer modeling technique or computer design. In this case, the drug design or ligand design is the design of molecule that will bind tightly to the target, or we can say it have a specificity to a particular target. The drug design is based on the knowledge of the three-dimensional structure of the biomolecular target, known as the structure-based design. Because now we are trying to dope, or you can say we are trying to make a comparison, or what is the interaction we are trying to find out with the binder site or with the receptors. Purpose of the drug design is to improve the affinity, selectivity, and stability of the protein-based therapeutics, especially. The major consideration for the drug design is to predict the binding affinity, alteration of ADL, the drug absorption, bioavailability, metabolism, half-life, and minimize the side effect or toxic effects. Okay, then we come to the next, the site of drug action. So the drug may bind to any tissue particular, it may go to a specific cell, or it may go to a specific component. Like if we are telling we are targeting on liver, so it's a tissue. Now in liver, there are different type of tissue, maybe some carcinoma, hepatic tissue also. So which particular type of tissue we are using for the targeting, it is important. Like if we are targeting any cancer tissue, so we generally use an approach of anchoring, that is a addition of or linkage of the vitamin H biotin to the particular drug, or in the nanocarriers we do. In case of molecular, we can target a specific type of receptor or any internal organ.
Sir, I think there's a technical glitch from your end. Sir, are you there?
He is not visible actually.
Yes, sir. As I can see that maybe he is facing some technical glitches and his microphone is not working in this case.
Correct.
I request the participants to please wait for a few minutes and let me get connected with him so that we can continue the session. I request, and I'm apologizing for the inconveniences caused. Please wait for a few minutes. We are sorry for the inconveniences caused, and it will not be possible for Dr. Sharad Bis to join or continue the session as he is genuinely facing some technical issues now. So to conclude with this session, may I now request Professor Dr. Dharmendra Ahuja, Professor and Dean of Jayoti Vidyapeeth Women University, Jaipur, to conclude the session. Sir, over to you. Sir, are you there? Dr. Ahuja, can you hear me? Please kindly confirm that whether I am audible to all of the participants. Can you please let me know? Okay, thank you, Urvashi. Dr. Ahuja, can you hear me? Okay, I think probably we are facing a problem where we can connect both of them. I just request you to hold a bit for a few minutes again.
Hello, Priyanka ma'am, can you unmute Dharmendra sir?
Yes, Debojyoti sir, I have already done from my end. I have. Dr. Ahuja, try to unmute yourself from your end as well. I think you can, sir, right now. Sir, is it possible for you to unmute yourself? I think he is facing some technical glitch as well because it's already.
Sorry for inconvenience.
Yes, sir. We were actually waiting for you. So
Yes, doctor, thank you. I'm showing my slides. Who's disabled? Doctor, I'm unable to connect or sharing my screen.
Sir, definitely, I'll make you the co-host in this case once again. Just 1 minute,
Thank
sir.
you. Thank you.
So check whether you are able to do it right now.
Yes, doctor. Thank you. Thank you, doctor.
Thank you. Thank you, sir.
It was a really informative session.
Okay.
Yeah.
Doctor, thank you. Thank you, doctor.
Yeah. Thank you.
So in this case, we can see if we are taking a 5, 6 and the 6 ring system, it will have a different type of expression. Or if we have this type of, we can say change in the 1, 2, 3, 4, 5, 6, I think octagon ring is this. So it will have a different type of regions also and interaction also. Similarly, when we when we have this structure like you can see in the n single bond n and here n single bond n, we increase the carbon also, and this structure is modified. Now this CO that was not interacting with another sites, now it is interacting with that also. So like this, it will affect the protein binding. These things generally we do in the in silico method, but once we synthesize, then we have to find out or prove it whether it is really working or not. In case of the computer drug designing, there are two major types of the drug designing that must be used for the
Sir,
drug
I'm
ligand.
extremely sorry for the interruption, sir.
Yes, please.
Actually, the participants have written in the chat box that they are unable to see the slides. It's becoming
Oh.
blurred in their case.
Okay, I'm again sharing it. Okay, okay, doctor.
Sure, sir.
Thanks. Yes, doctor. Is it okay now? Can you please confirm?
Sir, actually, it is visible, but the writings which are there on the slide, they are not properly visible. They are
Okay.
blurred in the case. So if you can just
Okay.
close the session once and then you can again show the slide, I think. Sir, if it is fine, you can go on in understanding. They will be listening to you, whatever you are teaching them.
Okay, doctor. I think if I stop my video, that will be easier. Because
I think it's better, sir.
yes,
Okay.
doctor. Yes. Yes, doctor. Can you please confirm? Is it okay?
Yes, sir. Yes, absolutely. It's fine, right?
Okay, thanks a lot.
Thank you.
In this case, again, if we see the first weak figure where the O is there, it is interacting with the H bond. If we add methyl group here, then methyl group, one number of carbon is increased and it will not interact with this particular O, while with the H it will interact. Same in case of the third figure, if there's H bonding, hydrogen bonding is there, okay. But methyl or because some steric parameters are responsible for the repelling of the molecule, or there's a less interaction. Sorry. So in case of the computer-based drug designing, first we have to confirm the drug, what is the drug design. Then we have to make it in a structure. We can compare different structures, then test, and then we analyze. If we find the optimum compound, then it is ready, not for the candidate, but for the further clinical trials. But before that, there are certain physiochemical properties that it should meet. There's one more way that is called combinatorial synthesis, where we can say add or prepare a very large number of chemical libraries using 96 well plate. So in this case, you can see in the first step there are 3 molecules. Now in the next step there are 9 molecules. We are adding different we can say ligands also, maybe some linker or maybe some additional groups that will bind to a specific site. Basic link, oh my god, what is that? So in case of this drug display cycle, we have a compound we have to collect first, then we do the primary assay with a high throughput screening. We can identify molecule also, and depending on the NMR and the mass, we can first confirm the structure elucidation. Then we can do some assays whether it's suitable or not in context of the viability or toxicity or efficacy to a particular action. If it is not, then again we have to modify the molecule. Then again we have to do the same things, mean to say that absorption, ADME or toxicity, these things we have to confirm, or even the protein binding. If we find each and everything are suitable, or we can say generally it is very difficult to find a molecule that is suitable or meet to all requirements. But if it is within a limit, then we can select that molecule. There is one software that is generally used, that is ADMET predictor. This particular software is used. Actually, we are not using software. We are using some free softwares. We did some work on it. I will discuss it later. So in this case, first we have a primary assay for the drug. We confirm the target sites also. Then we do the general screening, like drug lead, a fragment like component we can take, or depending on the molecular weight, we can confirm whether it can be given by which route. As you know, there are different mechanisms of drug absorption. In pharmaceutics, we generally focus on this. So whether the drug molecule is absorbed by the active mechanism, passive filtration, different mechanism. So generally the molecule having a molecular weight, 600 Dalton are absorbed by the passive mechanism. So we have to confirm or focus on molecular weight also, and its miscibility or solubility with the hydrophilic and hydrophobic, both type of environment. Then if we synthesize the component, we will check it's for IC50 or EC50. Even in the computer software, we design molecules and check its activity, like IC50 value, that is inhibitory concentration 50%. Or we can predict the solubility, stability, or selectivity, things with the receptor also. After that, we do the SAR, structure-active relationship. And once it is done and we find some molecule that is good, so we do QSAR, quantity structure activity relationship. This is generally used to confirm up to what extent it is giving the activity. We do some docking also, then some ADME filtering, mean to say, depending on the structure, we add or delete the group. So its ADME will change. Like these activities are also done to develop a lead molecule. In pharmacy, if we talk about, because in the final year, whether in IIM Pharma, we have a projects. So we can, the best way is, if we select any drug, whether it's herbal or any, we can authenticate, then we can do the successive extraction. It means to say, we select the solvent from a non-polar to polar region. So we can prepare a different type of extracts. Before you choose any right method for the extraction, you must know what component you want to work on or you want to separate, which solvent is good, which extraction technique you are going to use, and this what are the interfering compounds. For example, if I talk about the curcumin extraction, so in certain articles, they use defatting first, and then they are using methanol. In certain, they directly use the methanol. So in the both extract, they have a problem to separate the oily component. So that's why the selection of the right method should be known. Then we check its activity. If the plant is known, it's well and good. Okay. Now, if it is not known, then we can randomly check it for different activities, because we have to find out the effective dose also, or whether it's doing some activity or not. Maybe this molecule is of no use. Then we have to identify the lead. And finally, we do the phytochemical screening. In your extract, what are the different phytochemical constituents? It may contain alkaloid, or maybe flavonoid, maybe some other components. So first you confirm this one different component that is present in your extract. Then you will go for the pharmacological activity. Or you can do one more thing. In this phytochemical screening, if you are finding maybe 20 chemicals, you can again purify or refine it. As much how you refine, the better activity you will receive and better molecule selection will be there. Then we go for a pharmacological activity or a screening, whether it may be randomly selected or it may be selected on the basis of the literature. And once this pharmacological activity is done, like ED50 and the acute toxicity, all these things are measured, then we can try to formulate. But before formulation, we have to do some pre-formulation studies. There are certain softwares and the website that we use, like ChemSketch and ChemDraw. These are the generally freewares available online. Then we use some, because we did one herbal-based modification or preparation of derivative. I'll show you this. In this case, first we selected a molecule. There's a herbal molecule, and we try to find out if we do the modification of the compound, so whether its activity will change or how much will change or what are the effects. Then, so this is a simple draw of molecule on the ChemSketch software. Then, then this is a molecule. So in this molecule, we prepare a file, then turn it to the SMILE file. These are the SMILE files. These files are SMILE files, and we predict different properties. Like you can see here, water solubility, is a anti-cancer, intestinal absorption, permeability, glycoprotein. These type of activity you can predict using a free software online. Then we use this OpenBowl software to convert these file formats to get this SMILE files. Finally, there is one software that is called PKCSM. This is a software. If you load this SMILE file here, it will give you a data like this. So it is a different structure. So if we add this particular fatty acid chain onto the side, so how much there will be change in the structure, we can confirm by these things. But the thing is, it doesn't mean these values are really true. You have to prepare, we can synthesize the component, and then you have to confirm it by a practical mode. So, okay. So these are particular sites that are freely available, and some project-based things, whether for the M.Pharm, for the graduation, can be done using this software. And these are really very good software. And in preparation of this, these are my team. So they actually help me in this process or this synthesis of these molecules. We synthesize 64 molecules like this, and still the work it's going on it. Okay, so thank you. And if you have any query, I'd love to answer the questions. Hello, am I audible?
Yes, sir, you are audible. So thank you, sir. Can we expect some of the questions in the chat box from the participants? If you have any questions, can we have those? Yeah, we have already one question. That yes, what are the databases for molecules or drugs which can be used for docking against proteins, and the software which can be used to dock the whole library?
Whole?
Library of drugs.
Doctor, actually, the thing is, I belong to pharmaceutics. I'm not a chemistry person. First thing is this, but the thing is, whatever we can say learned by some of the softwares, not all softwares. So just wait a minute, I tell you. There are some docking. Autodock software is best. Then there's one version of this 1.57 that's freely available. I use this, and Pyrex also I sometimes use Pyrex. So I found Pyrex is quite easy as compared to the Autodock.
Okay.
Yes, doctor.
Thank you, sir. Next, we have the question is, what is the way to discover a new drug molecule, or what is the way to find the new thing in pharmaceuticals?
So in this case, as I discussed, nanotechnology is so much booming nowadays. First thing, first thing is this: to decrease the dose size and decrease the toxicity, improve bioavailability, and target specific things. Second thing, if we do not have any new molecule, we can find any new molecule by the what we say from the any resources and try to refine it. Like this molecule was the quercetin. I worked on quercetin molecule, but there was some issue with the solubility. So I was trying to figure it out using different type of liquids, or we can say the acids also. So in this way, if we design molecule is better. Even there are some synthetic approaches also, but those are very costly and chemicals are very costly. So better we make a design first, confirm the activities, and then we go for a synthesis.
Right. So moving to the next question ahead, we have one question that can you please let us know that what is molecular docking about?
Molecular docking. It is the, we can say, what is the interaction of the molecule or drug molecule or API to the particular tissue, or maybe it's a protein, it may be a receptor. That is basically the molecular docking. So whether what is the IC50 value also, and some surface free energy concept is also there. If it is minimized, so we select generally that type of molecule.
Correct. We have another question with us. Is there any software to know the lead from molecules?
Lead molecule? No, doctor. The thing is, if we have, suppose there are so many plants and some are still not explored, right? So the thing is, if we go for any trip, suppose you are going in a jungle forest, you can bring anything and then confirm what is this plant, then do the extraction isolation, then what is we can say this plant belong to which family. You can investigate the type of compound that is present there, and accordingly you can proceed.
Right. So we have another topic with us. I don't get the full question from Anjali Kadam. She has written that a virtual screening of drugs. Was this an answer or a topic asked by her? Can you please find the query regarding?
Can you repeat, please?
Virtual screening of drugs.
Virtual screening?
Yes.
Doctor, sorry for this question because I'm not so expert in this. So sorry. Yes.
Okay. Moving on to the next question, we have another. What kind of databases do you use for plant-based molecules? Duke database or PubChem?
No, doctor, I'm not so much deep with all these things because, I said, so I'm not sure because, the main thing is the technique or skills. The data may be so much, you can't remember all the data. But the thing is, if you have some names, so you can use those things. So I just use these two softwares. So I have more knowledge about these two actually.
Right. Yes. So thank you, sir. Do we have any questions from the participants? I feel like you can drop your questions again in the chat box and we'll answer back the questions from the speaker as well. Okay. So thank you, sir. Thank you for your session. I think that this session was quite an interactive one. And yes, definitely these docking molecules or the lead molecules which you are talking about, this comes exactly from the base where we are teaching the students from the bachelor's level. This goes up in the specialization of masters in pharmaceutical chemistry as well. And yes, they can find interest if they wish so, and they can move on to the Schrodinger, as it was told at the software, the PubChem, the ChemDraw you mentioned, or the ChemSketch. These are all the softwares, and I think that these basics will actually help the participants to find out their interest in. So we have with us right now Professor Dr. Dharmendra Ahuja, Professor and Dean of Jayapati Vidyapeeth Women's University, Jaipur. So, sir, I would now request you to conclude the session.
Thank you, ma'am. Thank you.
Sir, is it possible to you to please conclude the session? Dr. Ahuja? We request you to please conclude the session, sir. Say a few words about the session.
Yes, sir. Uh, पर वॉइस तो आ नहीं रही है.
अच्छा आप भी थे वो
वॉइस आ रही है सर.
Yeah, am I audible?
Yes, sir.
Yeah,
You
yeah.
are.
Yeah, actually there was some technical issue, so I was unable to listen the voice. Yeah. So means I hope I am means yeah, actually it was really informative session, and from morning onwards I have seen different session in morning. First of all, I would like to congratulate Adamas University, Honorable Vice Chancellor of the university. In fact, the founder and the Chancellor of the university, Dean Sajal Kumar Jha sir, then of course the convener Dr. Deepak Sharma sir, and Priyanka ma'am, who is handling the sessions, to all of the participants, to all the dignitaries who have delivered their session, to all the chairs. Means it was a really informative session altogether, and I congratulate all the stakeholders associated with the conference, and I wish a great success for the conference. Thank you. Am I audible?
Yes, sir, you are, and thank you so much.
Yeah.
We find your words as well as inspiring, and later, hopefully, that you also join us as a speaker. We look forward in that.
Sure. Yeah.
Thank you, sir.
Thank you.
Okay. We have come to the
Hello?
end of 4
हाँ मेरा
technical
फोन
sessions.
मीटिंग में बिजी है, ये जितेंद्र वाला है.
And, sir,
हाँ बोलिए. अच्छा हो गया ना. ठीक ठीक. आप ना वहाँ पे कुछ बोलिए मत उसको, नहीं तो फीस क्लियर नहीं करेगा ना वो लोग.
sir,
तो फीस का
can
पहले
you
क्लियर
kindly
करवा.
mute yourself?
हाँ आप फीस पहले क्लियर करवा लीजिए ना, उसको अमित शर्मा के पास भेज दीजिए. ठीक है.
Okay. In this case, what I would request is we are going to start very shortly the valedictory session. But before that, we have some of the videos from our stalwarts. So I would request Mr. Debojyoti Haldar, Assistant Professor of School of Medical Sciences, to kindly present few of the videos as we have received from various stalwarts across the world. Debojyoti, over to you. Kindly share your screen and let us all visualize it. Thank you.
यहाँ पे भरोसा नहीं, लेंस पे गए हैं.
Debojyoti, are you there?
Yes, ma'am, I'm here. I'm just sharing. Wait.
Sure. We are waiting. Thank you.
Hello everyone, I'm Ms. Sana Sizwan, Research Assistant from Faculty of Pharmacy. I would like to give a thanks for this nice presentation and this nice conference. It was really informative, and a special thanks to Dr. Devika Sharma for this invitation and opportunities. From Tajik International University. Hello everyone.
मैं दिखाई दे रहा हूँ।
Hi, I'm Shiva Nadeer. I'm from Jammu and Kashmir. I'm a research scholar pursuing PhD in pharmaceutical chemistry in the University of Kashmir under the supervision of Mubashir H. Masoodi. I had participated in third international conference on new horizons in drug discovery and development process, which has been organized by School of Medical Science, Adamas University, in collaboration with AICTE New Delhi. I would like to say that the conference was very beneficial and it really increased the research expertise and the knowledge of my research. And I would like to thank the organizing committee and the sponsors of this conference for conducting such a fruitful conference. All the technical sessions were very informative and it was really beneficial. It helped us a lot and it greatly enhanced our vision of research. I would once again like to thank them to give me an opportunity to show my poster presentation and some of my research aspects of my PhD. Once again, thank you so much. Hi, I'm Shiva Nadeer. I'm from Jammu and Kashmir. I'm a research scholar pursuing PhD.
Thank you so much. We have come to the end of today's session, and we have been waiting where we would like to present a token of appreciation to our last speaker, Dr. Visht. So can we request Debojyoti Halda to kindly present the certificate? Debojyoti. Thank you. Thank you, Debojyoti.
Thank you. Thank you. Thank you.
Thank you, sir. Thank you for your gracious presence. We have already shared the links with all of you. And I request both of you, both the links to be filled out for the participants in each case. And the last thing is that we will be declaring our results for the oral and poster presentation from 4 p.m. So I would request all of you to kindly wait till 4 p.m. or you can again rejoin at
Hello,
4
Manjula,
p.m. at the same time.
please unmute your. Hello.
Yes.
Ma'am, please unmute Manjula Pire. Your sound is coming.
I am unable to do that because she is constantly being muting herself. Okay, I think it's done from her end. Okay, thank you. So I think that when we are going to progress with the final session of declaring the results, it's been 4 p.m. So we would request you to wait till 4 p.m. through the same link, or you can again join back at 4 p.m. using the same link to hear the results being declared by our team. And one more thing, it is a kind request to all the participants to kindly see that you have filled both your feedback forms from the morning session as well as in the afternoon session, and wish you all the best in your future. Great to see you ahead in many such conferences and seminars to be held by Adamas University in future. Thank you. We conclude the session here, but again, I would repeat, I request each one of you to join at 4 p.m. for the declaration of the results for oral and poster presentation. Thank you so much.